Welcome to our session. Today, we'll be discussing the chapter Excretory products and their elimination. We already have studied that the cells are continuously producing energy by oxidation of food materials. But along with that, you have some byproducts produced in the cells, in the tissues. These byproducts include the carbon dioxide, water, etc. If carbohydrates are undergoing metabolism. Also, when nucleic acids, proteins are undergoing oxidation, nitrogen in them is removed in the form of ammonia. This ammonia is a dangerous product. Why it is dangerous? Because it is highly soluble in water of the cytoplasm and can affect the changes in pH. Because ammonia, once it dissolves in water, forms a very strong alkali, ammonium hydroxide. It is having high diffusion capacity, high solubility. So this should be immediately removed. So these waste materials are to be removed out of our body. Carbon dioxide is removed during breathing. When you are expiring, along with that air, we are sending carbon dioxide. Water is essential for our body. So the water produced can be used for body activities, whereas this ammonia and other nitrogenous waste materials that are produced. What is the nitrogenous waste material? Waste material in which nitrogen is present. That is to be removed, and their removal is called excretion. We also have some materials that are to be sent out of the body, which are all present in the body that enter through the food. We are not taking what is actually needed for our body. We will be taking excess of food materials than what we actually need. Some materials cannot be digested and absorbed. They are also to be sent out. So these products which are all produced in the body plus materials that are all ingested in excess, they are to be completely sent out of the body. And sending out of nitrogenous waste material like ammonia and other products from the body is called excretion. And for excretion, we are having different mechanisms. Ammonia is the first product. This ammonia is toxic and no problem, our body can send it out. The only thing is that it can be sent out in the form of a dilute solution. So it requires a lot of water to be gone out of the body. Along with water, ammonia can send out. It is not at all a problem for aquatic animals because they are living in water to the surrounding water. They will be diffusing. In simple organisms, there is no need of a special structure for sending out ammonia because as and when ammonia are produced in the cells, they will be sent out. They will be diffusing out to the surrounding water. So it is not at all a problem in lower invertebrates. Whereas in higher, especially triploblastic animals, the middle layer, the mesoderm and its derivatives are not in direct contact with the surroundings. So waste materials cannot be sent out directly to the environment. So in such organisms, there is a need of special structures to send out waste materials. And such organs which are concerned with sending out of nitrogenous waste material are called excretory organs. Ammonia will be sent out in aquatic animals, which can afford loss of water from the body. And that mechanism of sending out of waste material in the form of ammonia is called ammonotalism. And such animals which send out waste material in the form of ammonia itself are called ammonotelic. So most of the invertebrates, aquatic amphibians, aquatic insects, fishes are all ammonotelic. Okay, so the process of excreting ammonia in the form of ammonia itself is either through the gills surface as in case of fishes or through the body surfaces as in the case of small invertebrates. And in fishes, kidneys are not playing any role in its removal, because when the water is going out during breathing, through the gills, water is sent out continuously. Through that outgoing water, ammonia will be sent out. But for terrestrial animals, they cannot lose so much of water. Their problem is how to conserve water in the body. So in them, ammonia cannot be sent out as such. They are to be converted into other products which require smaller amount of water for their elimination. That's why ammonia is converted into urea or uric acid. If nitrogenous waste material is sent out in the form of urea, then mechanism is called ureotelism, and such animals are called ureotelic. We, that is mammals, adult amphibians, are all ureotelic. Cartilage fishes are also ureotelic. There, ammonia is converted to urea, and that urea is sent out in the form of a concentrated solution. So with minimum amount of water. That is the advantage of ureotelism. Less amount of water, waste material can be sent out. That is the adaptation for conserving water. Second thing is that urea is less toxic. So it need not be sent out immediately. It can be stored for some time in the body, no problem. It is soluble and can be stored in the body for some time. So in other animals who cannot even afford the loss of small quantities of water, just like insects, reptiles, birds, there ammonia is converted into the least toxic chemical called uric acid. Uric acid is least toxic among these three, and it can be sent out in the form of dry pellets. Uric acid is insoluble in water, so it can be sent out in form of dry pellets. So with very minimum or no loss of water, uric acid can be sent out. So it is an extreme case of conservation of water with no loss of water. Nitrogenous waste material is sent out in the form of uric acid. Uric acid is least toxic. So even if it is not sent out of the body, no problem. It is insoluble and will not interfere with the metabolism of cells. Such a mechanism of sending out of nitrogenous waste material in the form of uric acid is called uricotelism. And such animals are called uricotelic. So birds, reptiles, insects are all classical examples for uricotelicism. So in organisms, we are having ammonotelism, ureotelism, uricotelism. Okay. Now, different animals are having different organs for sending out of waste material. In the case of most of the invertebrates, these excretory structures are in the form of simple tubular structures called nephrons. They are in the form of simple tubular structures called nephrons. And protonephrid is the most simple, primitive type, which is also called flame cell. Flame cells are present in platyhelminths, some annelids, and in a group of chordate called cephalochordata, example Amphioxus, where waste materials are sent out. And in protonephridia, the structure consists of a coiled tube, which are mainly concerned with sending out of excess ions and water so that body will maintain a proper and constant ionic and water balance. The maintenance of a constant and steady water and salt balance in the body is called osmoregulation. So their main function is osmoregulation. Nephridia are tubular structures which are present in annelids and other invertebrates. They help to remove nitrogenous waste material and also maintain a ionic and water balance in the body. In insects, in cockroach we have studied malpighian tubules. These malpighian tubules are sending out nitrogenous waste material in the form of uric acid. So this uric acid is sent out as crystals or pellets, and they also help in conserving water, so regulation of water. In crustaceans are including crabs, lobsters, prawns, etc. They are having green glands or antennary glands. They are all aquatic. They have green glands or antennary glands that perform the function of excretion. So understand that different animals are having different types of excretory structures. Okay. In vertebrates, we are having kidneys. All vertebrates are having kidneys. And our kidneys are a pair of bean-shaped structures which are located on the thoracic cavity. To be correct, located between the last thoracic vertebra and the third lumbar vertebra. That is the correct position. Last thoracic. We are having 12 thoracic vertebrae. So last thoracic is also called 12th thoracic and third lumbar vertebra. Lumbar vertebra is the vertebra of the abdomen. Okay. Close to the dorsal wall of the abdominal cavity. Having a size of 10 to 12 centimeter in length, 5 to 7 centimeter in width, 2 to 3 centimeter in thickness, and will be weighing about 120 to 170 gram. Each kidney is a bean-shaped structure with a convex outer and a concave inner part. On that concave inner surface, there is a notch called hilum. Through this hilum, the renal artery is entering in. Renal vein is coming out. And the duct of the kidney called ureter is also coming out. The ureter will be opening into the kidney by means of a wide funnel-like structure. This wide funnel-like structure is called pelvis. The ureter is opening into the kidney by means of a wide funnel-like structure called pelvis. Kidney is covered by a tough, fibrous connective tissue capsule. And kidney has two parts, an outer cortex and an inner medulla. In the medulla, you are having some conical pyramidal shaped structures called medullary pyramids. These pyramids are directed towards the pelvis and they are all located in the medulla part. Pelvis is extending in between the pyramids and these extensions are called calyx, plural calyces. They are called calyx. In between the pyramids, the cortex is extending out, growing inward in the form of column called renal column or column of Bertini. Column of Bertini. To the surface of the pyramids, the tubules that brings urine opening. Okay. Now, the ureters are emerging out of their respective kidneys and they are all opening to a common chamber called the urinary bladder. Urinary bladder is opening out through a urethra. Urinary bladder is opening out through the urethra. It is through the urethra that urine is going out. Urinary bladders are opening separately to the urinary bladder. Okay, so that is the structure of our excretory organs. So kidneys, ureter, urinary bladder, urethra. Okay, now each kidney is made up of a large number of very tiny tubes, highly coiled tubes. Each tube is about 3 centimeters long. A million of them will be present in each kidney. They are the structural and functional units of kidney. They are called nephrons. Each nephron has two parts. One, the glomerulus. Two, the renal tubule. What is glomerulus? Glomerulus is a knot of fine capillaries, a network of fine capillaries, which is remaining inside the first part of the renal tubule. The first part of the renal tubule is a cup-like double-walled structure called Bowman's capsule. Actually, it is the Bowman's cup or Bowman's capsule, which is a double-walled cup-like structure. Now, it is within the cavity of that cup, glomerulus is kept. So, glomerulus along with the Bowman's capsule together is called the Malpighian body or renal corpuscle. These two together is called renal corpuscle or Malpighian body. Okay, this is the first part of the renal tubule. The remaining part is in the form of a highly coiled tube, which is having 3 distinct parts. The first part, very close to the Bowman's capsule, is highly coiled and so is called proximal convoluted tubule. Then it is followed by a hairpin bent structure called loop of Henle or Henle's loop. This Henle's loop is having the first descending limb and then ascending limb. Both descending and ascending limb will be having a thick limb and a thin limb. Thick and thin. Okay. Now, the ascending limb of Henle again become highly coiled and that second coiled part is called distal convoluted tubule. Distal convoluted tubule of several such renal tubules open to a straight tube called collecting tubule or collecting duct. It passes deep and deep into the medulla and open to the surface of the pyramids. Okay, so connecting ducts are opening to the medullary pyramids, on the surface of the medullary pyramids. This is the part of a nephron. Remember, our entire excretory system is developing from mesoderm. Our kidney is of metanephric kidney, which is also called head kidney. Metanephric or head kidney. The most developed type is metanephric. Sorry. It is also called, it is tail kidney, because it is from the posteriormost part, that posteriormost part of the nephrotom part, that our kidneys are developing, so it's called tail kidneys. So metanephric and tail kidney. Okay. Of these parts, the Bowman's capsule along with the glomerulus, PCT and DCT may be lying in the cortex, whereas in some nephrons, the Henle's loop is long and well developed, and they may go deep into the medulla. And depending upon the nature, we are classifying nephrons into two categories, namely a cortical nephron and medullary nephron. Actually, it is called juxtamedullary. Why it is called juxtamedullary? Very close to medulla. That's the meaning. Juxtamedullary nephron. In cortical nephron, which is most abundant, 85% of our nephrons are belong to this category. The glomerulus, Bowman's capsule together is called the renal corpuscle. So the renal corpuscle, proximal convoluted tube, distal convoluted tube, and the Henle's loop. Henle's loop is very small. They are all lying in the cortex. Juxtamedullary, on the other hand, is only a very small percentage, 15% average, and they are lying just above the medulla, close to the medulla. That is why it is called the juxtamedullary nephrons. And here, the renal corpuscles, PCT, DCT, are lying in the cortex. Whereas Henle's loop of Henle is long, well developed. They are growing deep into medulla. They will be extending deep into the medulla. Clear? So, these are the two types of nephrons in our kidneys. And it is the juxtamedullary which are playing an important role in concentrating our urine. Our urine is made concentrated. Not only our kidneys are filtering urine and sending it out, but is sending it out in the form of a very concentrated urine. That concentration is done by the juxtamedullary. How it is capable of concentrating, that is what we are now going to study. Okay. Now, kidney is not producing urea. Urea is produced in liver in a chemical reaction called Krebs-Hansley cycle. Liver is converting one molecule of carbon dioxide, two molecules of ammonia, and three molecules of ATP into a molecule of urea. Okay. This is through a cyclical reaction called Krebs-Hansley cycle, or simply ornithine cycle. Through a cyclical event called ornithine cycle, the ammonia, carbon dioxide, and ATP are used to produce urea. This urea is water soluble, so it can be carried through the blood. So carried through the blood from liver, it is brought to the kidney. Kidney is receiving large quantity of blood. In earlier chapters, we have studied that our ventricles are pumping about 5 liters of blood every minute, and the kidney is receiving almost 1/5 of that blood. That means kidney can receive 1100 to 1200 ml of blood per minute. Okay, renal artery is bringing blood. This renal artery, after entering into the kidney, branches with a number of arterioles, small branches called arterioles. And a small branch of arteriole called afferent arteriole. This afferent arteriole is bringing blood to the glomerulus. Okay, so blood is brought to the glomerulus through afferent arterioles and are taken away by efferent arterioles. After coming out of the glomerulus, the efferent arteriole again branches. This efferent arteriole again branches and will be forming a fine network of capillaries around the PCT, DCT, etc. Since this capillary is around the tubule, we call it as peritubular capillary. Peritubular capillary. And one branch of peritubular capillary will be growing close to, it is growing close to our Henle's loop. That is called that blood vessel is called vasa recta. So vasa recta is well developed in juxtamedullary nephrons, whereas this vasa recta is poorly developed or even absent in cortical nephron. Okay, so you are having a vasa recta well developed in juxtamedullary type. It is a part of the peritubular capillary. So this peritubular capillary will be again fusing to form a vein, and these veins join to form renal vein that takes blood out of the kidney. So kidney is receiving 1/5 of the blood. And what is the route? I will repeat once again. Renal artery, arterioles, afferent arteriole, glomerulus, efferent arteriole, peritubular capillary, veins, renal vein, outside the kidney. As blood is passing through, filtration is done by the kidney. That is what kidney is doing. Kidney is filtering materials that are to be sent out of the body from blood. That is the first event that is occurring in urine formation. There are 3 events. The first one is ultrafiltration. Why it is called ultrafiltration? Remember, the blood is passing through the glomerulus under high pressure. The pressure will be almost double the pressure present in any other capillary. See, the arrangement of afferent arteriole and efferent arteriole, you can see from the diagram. This is large. More blood is coming. Less blood is taken out. So, there will be more pressure in the glomerulus. It is double, 60 mm of mercury. That is the pressure present within the glomerulus. And under that pressure, the certain special situations will help in filtering soluble materials to come out of the blood. What are these special conditions? Remember, the blood is separated from the cavity of the Bowman's capsule by 3 layers. 1. Endothelium of the capillary. Endothelium of glomerulus. This glomerulus is having a number of small gaps called fenestra. Fenestrae plural. Okay. This is the only layer present in the walls of the capillary, endothelium. And the inner wall of the Bowman's capsule, that is having epithelium. That epithelium is having some specialized cells. These specialized cells are having a shape similar to that of our foot. So it's called podocytes. Though the podocytes are arranged close together, it is an epithelium. Okay, inner epithelium of the Bowman's capsule. It leaves between them some gaps. These gaps are called slit pores. These gaps are called slit pores. And the two are connected together by the basement substance. Basement substance. So, a 3-layered filtration membrane separates the blood flowing through the capillary and the capsular space. This is the capsular space. And there are 2 conditions. What are they? Blood is under high pressure and the membrane is highly porous. You are having slit pores or filtration slits and fenestrae on the membrane through which the liquid part of the plasma will be coming out. Larger proteins cannot come out because of the larger size, but all the soluble minerals, amino acids, glucose, that is essential materials, plus non-essential materials of the plasma, like urea, water, etc., will be also coming out. This filtration is thus called ultrafiltration because almost everything in the plasma is coming out. Blood cells cannot come out because they are large in size. Okay, large proteins cannot also come out, and they get accumulated in the cavity. This cavity is called capsular space. The first fluid thus filtered from blood is called glomerular filtrate or primary urine, which is having the same osmolarity, that is salt concentration, as that of blood. Okay, this first urine or primary urine become accumulated the capsular space, that is carried through the PCT and then Henle's loop, DCT, etc. So this is the first event called the ultrafiltration. Is it clear? Ultrafiltration. Podocytes are arranged in an intricate manner so as to leave between them some small minute spaces called filtration slits or slit pores. And blood is filtered so finely through this membrane that almost all constituents of the plasma, except the large sized proteins, pass into the lumen. It is called ultrafiltration. Is it clear? Now, the second event is occurring as the urine is passing through the tubule. Third event is also taking place along with that. I told you that kidney is receiving about more than 1 liter of blood per minute. Out of that, kidney can produce 125 ml of urine per minute. This is called glomerular filtration rate. The rate at which filtration is occurring, the amount of filtrate formed by the kidney per minute, that is called glomerular filtration rate. So, out of 1100 to 1200 ml of blood passing through the kidney in every minute, 125 ml is filtered out. Okay, so almost 10%. So 125 ml per minute means 180 liters per day. So our kidney is capable of making so much of amount of urine, but we never send out 180 liters of urine per day. We hardly send 1.5 to 1.0 to 1.5. That is the maximum amount of urine that we are all sending out. So what happens to the remaining? That is what now we are going to study. As the urine is passing through the PCT and DCT, there occurs another phenomenon called tubular reabsorption. What is that tubular reabsorption? As the primary urine is passing through the PCT, all the essential materials like amino acid, glucose are actively absorbed, completely and actively absorbed back. All the nutrients are absorbed back. Now, there is absorption of bicarbonate, sodium chloride, etc. 70 to 80% of electrolytes are absorbed back. See, the PCT and DCT are surrounded by peritubular capillaries. Into that peritubular capillaries, the walls of the PCT and DCT. They are made up of cuboidal epithelium. They are all actively involved in absorbing these materials needed for our body back into the blood. This is a selective process. Tubular absorption is a selective process. It is selectively taking only those materials that are needed for our body are absorbed back. That's called selective reabsorption of materials needed by a body as the urine is passing through the PCT. What are the materials that are absorbed? All the nutrients like glucose, amino acids. See, glucose and amino acids are also coming because the filtration is a non-selective one. Everything in the plasma is filtered out. From that, essential materials are absorbed. This absorption also takes place in DCT. Water absorption, salt absorption like sodium chloride, bicarbonates are absorbed actively back into the body, that is back into the blood. But the absorption that is taking place in the DCT is depending upon their requirement in the body. That's why it is called conditional reabsorption. Reabsorption taking place in the DCT is depending upon their requirement in the body. Suppose our body requires more sodium, then more sodium is absorbed. If our body doesn't require any sodium, no absorption will be taking place. Similarly, water. If our body requires more water, then more water is absorbed here. So, this absorption taking place here is called conditional reabsorption. So, essential materials are absorbed back through the PCT and DCT. That's the second event. And third event is tubular secretion. This tubular secretion is also selective, meaning those materials that are to be sent alone, those which are to be sent out will be alone sent out. From where? Here you are having peritubular capillaries. From that peritubular capillaries, materials that are not passing through this filter but should be sent out of the body are sent out by the cells of the PCT and DCT. They are all secreted into from the blood. Here you are having the peritubular capillary. From that peritubular capillary blood, materials are separated and sent into the lumen. What are the materials that are thus sent out? You will be having materials like hippuric acid, creatinine, penicillin. See, these materials cannot pass through the filter, but our body should send it out. Then hydrogen ions, potassium ions, and this is the potassium ion. Ammonia, small quantity of ammonia are dissolved in the fluid by the tubular secretion. These materials are sent into. Here also there is tubular secretion. Here again it is conditional. Depending upon the balance in the body, sodium and potassium balance. See, either more sodium will be absorbed or more potassium is released, thereby maintaining the ionic balance, sodium potassium ion balance, thereby keeping the pH of the body fluids also stable. Also, you will be having bicarbonates absorb negative charge and positive charge hydrogen ions secreted. to variable levels so as to maintain the pH of the body. So ionic balance and pH balance are maintained. Water is also absorbed, thereby maintaining a constant and steady water level. Okay, so kidney is not only helping in sending out nitrogenous waste material, but also is helping in maintaining ionic balance, pH balance, water balance. Maintenance of water balance is called osmotic regulation, and maintenance of salt and pH balance is called ionic regulation. Osmotic regulation plus ionic regulation together is called osmoregulation. Kidney is having an important role in maintaining water level, salt level, and pH level in our body. Clear? So, understand that tubular reabsorption and tubular secretion are selective. It occurs as the urine is passing through the different parts of the tubule. So, we are having a total of 3 events in urine formation. Urine is the solution contain urea that is produced by the kidney. Urea is the main ingredient that is produced in the liver. Understand the difference? Urea is the compound, chemical compound. Urine is the solution. So, ultrafiltration, that is non-selective. Tubular reabsorption, selective. Tubular reabsorption, again selective. These are the 3 important events in formation of urine. Now, in urine formation and elimination, what are the role of different parts of the tubule? Let us study. Remember, the inner wall of the Bowman's capsule is made up of inner epithelium that is containing podocytes that help in filtration. In the wall of the PCT, you are having cuboidal epithelium, which is rich in mitochondria and microvilli. That is why it is capable of secreting and absorbing. Active transportation is possible because they are having abundant mitochondria. And to increase the surface area, you are having breast border epithelium. Microvilli containing breast border epithelium here. And they will be absorbing materials that are essential to the body and sending out materials that are not needed to our body. So by absorption and secretion, the PCT is mainly playing an important role in maintaining the osmotic balance of our body. The main function of PCT is to maintain pH and ionic balance of the body, fluid, thereby selective secretion of hydrogen ions and ammonia into filtrate and absorption of bicarbonate from it. So the main function is maintenance of a water and ionic balance. Whereas Henle's loop is having an ascending part and a descending part. The first part is the descending part. It is having a very short thick limb and a very long thin limb. Now, the ascending part is having a very thin limb and a very long thick limb. Both descending and ascending limb are having a thick and thin, but the relative length of thick and thin limbs are different. Thick limb of Henle is made up of cuboidal epithelium, but they are all very poor in mitochondria and microvilli. Whereas the thin limb is made up of, this part is made up of squamous epithelium. And since it is made up of squamous epithelium, it is permissible to water. The descending limb is permissible to water, but not permissible to ions. Whereas the ascending limb, see, it is permissible to the salts. Sodium chloride is escaping through the thin limb, and this transportation of sodium chloride is passive. Here it is passive because you are having only squamous epithelium, thin, which is permissible. So sodium chloride is entering into the intercellular space passively, whereas through the thick limb, sodium chloride is pumped out actively. So ascending limb is permissible to salt, descending limb is permissible to water. Clear? Ascending limb is having a very thin limb and a very long thick limb. Now, DCT is also made up of cuboidal epithelium, which is rich in mitochondria and brush border. Henle's loop is playing no role in osmoregulation, but playing an important role in maintaining a high salt concentration in the medullary interstitium. What is interstitium? Cells in the medulla are remaining bathed in the fluid, which is remaining within them or between them. That fluid which is present between the cells of the medulla is called medullary interstitial fluid. That is intercellular fluid. The intercellular fluid in the medulla is very, very salty in nature. How it is? That is mainly the function of Henle's loop. I will be coming to that point next. Now, DCT is mainly made up of cuboidal epithelium with brush border and abundance of mitochondria, which is concerned with absorption and secretion. So by absorbing sodium chloride back into the body, water, bicarbonates, and sending hydrogen ions and potassium ions, it is maintaining the salt balance and ionic balance. So it is playing, DCT is playing an important role in ionic balancing, water balancing, so osmoregulation. Now, what about the CT? CT is also lined by cuboidal epithelium. CT is passing deep into the medulla. This is the cortex, this is the medulla, and its walls are made up of cuboidal and are permissible to water. There are some cells which are highly permissible to water. So because of that, water escapes. As water is escaping, the urine becomes concentrated. It is the CT that makes the final urine, which is highly concentrated. The final urine is made concentrated 4 times than that of the primary urine. How? That is the technique of our kidney. See, kidney is having a high osmotic capacity or osmolarity maintained within the medullary interstitium. Okay, now I will be drawing a new diagram for you. Only Henle's loop and vasa recta are involved in that. Okay. This is the descending limb of Henle and this is the ascending limb of Henle. We studied that the thin limb of Henle is permissible to water. And ascending limb is permissible to sodium chloride, salts. Through the thin limb, sodium chloride is going out passively. And through the thick limb, it is pumped out actively. Because you are having cuboidal epithelial cells, here sodium chloride will be pumped out. And in maintaining a high osmolarity, the CT is also playing an important role. Collecting tubule is also playing an important role. Now let us see what is happening. The sodium chloride that is escaping from the ascending limb enter into the medullary interstitium, that is in between the cells of the medulla. Only diagrammatic representation. You are having the fluid called intercellular fluid, which is called interstitial fluid. In between the cells of the medulla, you are having the fluid. And all cells are remaining bathed in that fluid. It is called intercellular fluid, interstitial fluid. To that, sodium chloride is pumped out. The CT is also releasing small amounts of urea. Small amount of urea is released from CT. Remember, the presence of sodium chloride and urea in the medullary interstitium will make the salt concentration in the deeper part of medulla very high. Salt concentration will increase the ability of this fluid to absorb water by osmosis. So, osmosis is a function of, or osmosis depending upon the salt concentration in the fluid, correct? Which is called osmolarity. The osmolarity of medullary interstitium goes higher and higher as we go deeper and deeper. Why? More and more sodium chloride, more and urea. Both of them are now dissolving this fluid, and that makes the salt concentration high, so that osmolarity of the medullary interstitium is very high. It is to that medullary interstitial, or through the medullary interstitial fluid, that our blood vessel, what is the name of the blood vessel? Vasa recta is passing. Vasa recta is a thin capillary which is running parallel, through which the blood is flowing in a direction opposite to the direction of the flow of urine in the Henle's loop. Thus, you are having fluids flowing in opposite directions close together, which is called a counter current. Urine is flowing through the descending limb, then ascending limb in opposite directions. That forms a counter current. Blood is flowing through the descending limb of descending limb of vasa recta, and this is the ascending limb of vasa recta. This also is in a counter current form. And between the fluids of, that is the blood of vasa recta and urine of Henle, there is also another counter current system. So more than one counter current system is there, which is called counter current multiplier system. Now, sodium chloride that is escaping out from the ascending limb of Henle and urea escaping from the CT make the medullary interstitium highly concentrated. Thus, a concentration gradient, osmolarity gradient is generated in the deeper part of medulla. Meaning, in the deeper part, the salt concentration goes on increasing. In the outer cortex, it is just 300 milliosmol per liter. That is the osmolarity. Salt concentration in the fluid, that is in the blood or any other fluid there, it is just 300 milliosmol. But as you go deeper and deeper, it becomes increased to 600, 900 and finally reach 1200 milliosmol. That is the osmolarity of the medullary interstitium. It is through the medullary interstitium that this vasa recta is passing. As vasa recta is entering into the deeper part of medulla, the sodium chloride that is escaping from the ascending limb enter into the descending limb. I will repeat. Sodium chloride that is escaping from the ascending limb of Henle enter into the descending limb of vasa recta. And more salt is coming, meaning osmolarity of the blood in the vasa recta is also becoming equal to that of the osmolarity of the medullary interstitium. That means medullary interstitium is having 1200 milliosmol. So the blood here also becomes equal to the salt concentration in the medullary interstitium. It is this highly concentrated, salt concentrated blood whose osmolarity is 4 times than that of the cortical blood. Normally, blood will be having only 300 milliosmol osmolarity. Now, on reaching the deeper part of the medulla, become highly hypertonic. Its osmolarity increases to 1200. And as this thick concentrated blood is passing very close to the Henle, remember Henle's loop is highly permeable to water because it is made up of only squamous epithelium. The water escapes out and that water will be absorbed into the ascending limb of Henle. So more and more water enter into the blood. Why? Because blood is having more salt, so its osmolarity is very high, and because of osmosis, water enter in. And all the water that are entering into will make the osmolarity of the blood decrease as blood is ascending through the ascending limb of vasa recta. So osmolarity of the blood decreases to, say for example, from 1200 to 900, 600, and finally, as it reaches the cortical part, sodium chloride is also pumped out. The salt is also pumped out. As sodium chloride is pumped out, what happens? The salt concentration decreases. So, salt concentration decreases, water level increases. So, the osmolarity of the blood decreases and will reach again that 300 milliosmol as it reaches back the cortex. So, what actually has happened? Sodium chloride that escaped from the ascending limb entered into the medullary interstitium, made the medullary interstitium highly concentrated, and that sodium chloride was exchanged with the descending limb of vasa recta, that made the blood of the vasa recta also very concentrated, that caused water from the Henle's loop to be absorbed. Okay. Now, after absorbing water, it loses sodium chloride also in the ascending part of the vasa recta, and thus the blood again become same as that of the any other part of the body. That is isotonic with other parts, having only 300 milliosmol. So the net effect is that most of the water from the kidney, from the Henle's loop is absorbed and entered into the blood. Now, what happens to the urea, which is also increasing the osmolarity? Small part of this urea enter from the medullary interstitium. That urea is entering the ascending limb of Henle, and that urea goes out. Now understand, as water is flowing through the CT, through the medullary interstitium, outside the CT, the concentration is very high. Medullary interstitium is having a high salt concentration. So because of that, it will be absorbing water till the concentration of the urine inside and the medullary interstitium outside become equal. So salt concentration outside is 1200 milliosmol. The concentration of the urine inside the CT also become 1200 milliosmol. So till it become equal, all the water is escaping, and these water are taken by the vasa recta. The blood is absorbing, taking all the water back. Thus, by maintaining a high osmolarity gradient between the cortex and the medulla, most of the water, 99.9% of the water, remember, our GFR rate is 180 liter per day, but we are sending only 1 to 1.5 liters of urine a day. So what happened to remaining? They are all absorbed back. So most of the water, 178.5 liters of water absorbed back. How? By passive mechanism, osmosis. By simple mechanism of osmosis. For absorbing that much of water, no energy is needed. But for absorbing that much of water, we have to maintain a high osmolarity in the medullary interstitium. For that, salt is to be pumped out and small amount of energy kidney used. So by using a very small quantity of energy, it maintains a high osmolarity in the medullary interstitium with the help of the two salts like sodium chloride and urea, so that most of the water is absorbed by simple osmosis. So the urine that is finally passing out will be have the same salt concentration, 1200. The urine that formed first, primary urine, is having the same salt concentration as that of the blood, that is 300 milliosmol. The final urine that is going out will be having a concentration of 1200. That is why I told you that the urine is made 4 times concentrated. So our kidney is not only simply filtering and sending out the urine, but also making it concentrated so that most of the water is absorbed. Okay. Now, the urine passes from cortex to medulla, then from medulla to cortex, and from cortex to medulla. And whenever it is in the cortex, it is isotonic, and as nutrients are absorbed, it becomes hypotonic, meaning very dilute. The urine was initially isotonic in the Bowman's capsule, but when nutrients 70 to 80% of the electrolytes are absorbed, it becomes hypotonic, dilute. It is that dilute urine that is passing, and when it reaches the deeper part, it again becomes, because water is absorbed, it becomes hypertonic. Because water is absorbed, so it became concentrated. Now, salt is pumped out, again it becomes hypotonic when it reaches the cortex. And that dilute urine passing through the CT, water is absorbed, again becoming hypertonic. So, hypertonic, 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 hypertonic. Water is absorbed here, water is absorbed here. This is the mechanism by which our urine is made 4 times concentrated. That is with the help of the Henle's loop. Henle's loop is having a big role in maintaining high osmolarity gradients, and it is because of the maintenance of a high osmolarity gradients that most of the water is absorbed and urine is made concentrated. That is the role of Henle loop. So we are having counter current mechanism, fluid passing through opposite directions in closed by tubes. For urine also you are having a counter current mechanism. For blood also you are having a counter current mechanism. And between the blood flow and the urine flow, there is also a counter current mechanism. Because of this, finally, our urine is made 4 times concentrated than that of the primary urine. Okay. So, this is the mechanism. Urine is passing through the CT. Now, urea is escaping, that is entering into the ascending limb and then goes out. Sodium chloride, that is escaping out, entering into the descending limb of vasa recta, escaping out. Okay, so we are having this mechanism operating continuously. Mammals have the ability to produce a concentrated urine, and Henle's loop and vasa recta play an significant role in this. Now you will understand what is the role of the Henle's loop. Henle's loop is having no important role in maintaining the water level, salt level, etc. So, reabsorption is minimum in this ascending limb. However, this region plays a significant role in maintenance of a high osmolarity of the medullary interstitial fluid. You got it? Now, how this flow is regulated and maintained? This is the diagram I have given you now. This represents the loop of Henle and this represents vasa recta. This vasa recta running parallel to that of this one. Clear? Now, who is regulating kidney functions? Kidney functions are regulated by 3 different hormonal mechanisms. Okay. 1. When there is a fall in blood level in the body. Why? Because we are losing a lot of water. When you are sending out dilute urine, a lot of water, blood become concentrated. That means its osmolarity increases from 300 to higher levels. Then our osmoreceptors present in different parts of the body become activated. So when there is an increase in osmolarity, when there is a dilute urine is passing or excess loss of water through urine, that will cause increase in osmolarity of blood. That will activate osmoreceptors. These osmoreceptors send the message to the hypothalamus of the brain. This hypothalamus is producing a hormone called ADH, anti-diuretic hormone, or oxytocin. This oxytocin or ADH has 2 actions, dual action. What are their actions? 1, it will act on DCT and will cause more water to be absorbed. So, it will increase permeability, increase absorption of water. Okay. When there is more water absorbed, urine becomes concentrated and more water comes to the body. That means the osmolarity of the blood decreases. Second one, this directly acts on blood vessels, walls of the blood vessels. That's why it is called oxytocin. Sorry, it is vasopressin. There's a correction, please. ADH is called vasopressin because it is pressing on the blood vessels so that blood pressure increases. Okay, so vasopressin is produced from the posterior lobe of the pituitary. Hypothalamus is producing this hormone which is stored in the posterior pituitary. And now from the posterior pituitary, it is released. It increases the body fluid volume. And when the body fluid volume, that is blood volume, is increasing, the osmoreceptors are suppressed. So ADH release the complete feedback mechanism. ADH can also affect the kidney function by its constrictory effect of blood vessels. It acts on blood vessels, causing the BP to increase. This causes an increase in blood pressure. And increase in blood pressure can increase the glomerular blood flow and thereby the glomerular filtration rate. So that is the second way in which ADH is. Why it is called ADH? It is against diuresis. What is diuresis? Sending out of dilute urine is called diuresis. This hormone prevents diuresis because it helps in reabsorbing water. So it is called anti-diuresis, ADH or vasopressin. Second one is through another system called JEE system. What is this JEE system? We have to study that part. We are having a endocrine secretory part formed between the wall of the afferent arteriole and the distal at the point where the DCT and wall of the afferent arteriole are coming and meeting each other, there is a thickening develop. That thickening develop here is called juxtaglomerular apparatus. This juxtaglomerular apparatus is having secretory cells called juxtaglomerular cells. These juxtaglomerular cells are present on afferent arteriole wall, on the wall of, this is the juxtaglomerular cell. It is secretory. It is secretory in function. Similarly, on the wall of the DCT, this is the DCT, there is a thickening where you are having the second type of cell called macula densa. This macula densa is having, when it is present on the wall of the DCT, it is sensory in function. It will detect changes in the salinity of the urine that is passing through it. What much of water is there, what much of salt is present, etc. If there is a fall in GFR or blood volume or blood pressure, immediately the macula densa understands and will activate JG cells of the juxtaglomerular apparatus. Okay, now this will secrete a hormone enzyme called renin. Spelling is RENIN because we have studied RENIN. This renin is activating a protein present in blood. Okay, we are having an enzyme which is actually produced by the liver, called angiotensinogen. It is angiotensinogen. This angiotensinogen is produced by liver. Angiotensinogen is an inactive gen, is converted into active angiotensin 1. Angiotensin 1 is converted into angiotensin 2. For that, there is another enzyme called angiotensin converting enzyme. This angiotensin 2 is having two functions. One, it will act on adrenal cortex and activate the production of a hormone called aldosterone. Aldosterone is a steroid hormone produced by the adrenal cortex, which will increase the sodium absorption back from urine, from alimentary canal, and also from the sweat produced by our skin. So sodium content in the blood is very much increased. When there is more sodium, what will happen? Osmolarity of the blood is increasing. And when the osmolarity of the blood is increasing, sodium absorption, it increases the osmolarity. Increase in osmolarity of the blood. That will increase more water to be absorbed. So, more water reabsorption. It will increase water reabsorption. Whenever more water is absorbed, blood volume increases. When blood volume increases, blood pressure also increases. When blood pressure increases, GFR is increasing. When GFR, blood volume, blood pressure are normal, that will be suppressing the production of renin. So there is no need of any more renin. So renin production is suppressed. That is one way. This mechanism, see, angiotensin 2 act on aldosterone production, increases the GFR through the salt, through the sodium chloride. This is the main mechanism. Now, angiotensin, what is the meaning of that term? Tensin, increasing the tension, pressure. It is also increasing the blood pressure. It increases blood pressure because it is acting on blood vessels and causes increase in blood pressure. When increase in blood pressure is there, it will increase the GFR. When blood pressure is increased, blood pressure increase will cause more filtration to occur, and this also will help in suppressing the renin production. This is the second way of action. Clear? The whole mechanism is called RAS system. Renin, angiotensin, aldosterone system, RAS system. This is a mechanism, main mechanism by which the kidney is regulating its activities. Okay. Now, when you are having more water, more pressure in the blood, kidney will understand that there is more water, there is more pressure, GFR is very high. Now, its atrial wall will sense it and will cause the production of a factor called atrial natriuretic factor. Increase in blood flow to the atria of the heart causes the release of ANF. This ANF is a vasodilator. It causes blood vessels to dilate. This is the only one which can increase the blood pressure. Decrease the blood pressure because it's a vasodilator. blood vessels are dilating. Dilation of the blood vessel cause a decrease, a fall in blood pressure. And when blood pressure falls, GFR is also reduced. So, RAS is increasing the GFR, blood pressure, and ANF is trying to decrease. So, these two are all antagonistic in action. Thus, ANF mechanism acts as a check on the renin angiotensing mechanism. Clear? Now, when that urine is coming and filling the urinary bladder, what happens? The bladder becomes full. And as bladder becomes full, it becomes stretched. The walls are having stretch receptors. They will send this message of stretching, okay, to the central nervous system. So, stretch receptors on the walls of the bladder send signal to the central nervous system, and the central nervous system then passes on the motor messages. So that this motor message from the central nervous system, it is having CNS, having two action, where in the urinary bladder muscle, it causes contraction, and to the sphincter, that circular valve-like muscle around the urethra to dilate. So, it will cause dilation of the sphincter and walls of the urinary bladder to contract. So, urine goes out. This is called micturition, and the entire mechanism causing it is called micturition reflex. We will be sending out 1 to 1 liter of urine per day, and that urine is slightly acidic. Why? Because we are releasing hydrogen ions at the DCT. So, urine is slightly acidic in pH. pH is 6.0. It is having a characteristic odor because of the urinoid crystals, which become split up into ammonia, releasing ammonia. That is why urinals are smelling. They are having a pungent smell when it is not properly washed and cleaned. On an average, we send out 25 to 30 gram of urea per day. And this may be slightly different. We are not sending all the urea produced. Still, we will be having 17 to 25 milligram of urea per 100 ml of blood. That is the normal value. And the urine may be very often containing abnormal ingredients called glucose and ketone bodies. Presence of glucose is called glucosuria, and ketone bodies is called ketonuria, which are the symptoms of diabetes mellitus, which is simply called as sugar. So, in diabetic patients who are having sugar, that glucose is an ingredient that is passing out through the urine. In normal healthy man, there will be no glucose. All the glucose will be absorbed. Now, along with kidney, we are having some other organs, which are our lungs, which are removing carbon dioxide at the rate of 200 ml per minute. Similarly, our liver is secreting bile that is containing bilirubin, biliverdin. These are all excretory products. Okay, these are all actually waste products, broken products of hemoglobin, cholesterol, degraded steroid, vitamins, etc. Sweat is also producing. Sweat and sebaceous glands are produced by the skin glands that also eliminate some amount of waste materials. Sweat produced by the sweat gland contains sodium chloride, small amount of urea and lactic acid. And the sebaceous glands are containing substances like steroids, hydrocarbons, waxes, which is in the form of sebum. That oily secretion is called sebum. Similarly, saliva is also having some small amount of nitrogenous waste. So, in addition to the kidneys, our lungs, liver, skin and salivary glands are also acting as excretory organs. And finally, coming to the disorders when excess of urea accumulates in blood, the condition is called uremia. It is an indication of kidney failure. Then that excess urea can be removed by hemodialysis. And if hemodialysis is continued for a long time, that will be causing severe problem for the patient. In hemodialysis, blood is taken out of our body from a convenient artery and given to an artificial kidney, and the blood drained from the convenient artery, pumped into dialysing unit after adding anticoagulant like heparin. And what is this dialysing unit? It just consists of a coiled cellophane tube which is kept in a chamber, and the chamber is filled with a dialysing fluid, and this dialysing fluid will be having the same salt concentration as that of blood, except for there is no nitrogenous waste. So as our blood is passing through the tube by osmosis, only nitrogenous waste material is coming out to the dialysing fluid, because in the dialysing fluid there is no nitrogenous waste. In the blood there is high concentration, and the cellophane tube will be acting as a semi-permeable membrane through which they will be filtered. Okay, so as nitrogenous waste materials are removed, blood become again normal, and it is now pumped back to the vein after adding anti-heparin to this. Okay, when kidney transplantation is to be done, that is when this type of hemodialysis is to be continued, that is not a permanent solution. When renal failure occurs, we have to get a kidney from a donor. A functioning kidney is used in the transplantation from a donor, which is preferably a close relative. Why? If you take kidney from anybody else, it may be rejected out, because our body immunity will be fighting is mismatching. So there is a chance of that kidney transplant rejected out. To minimize that, we have to get it from a preferably a close relative. And the success rate of such complicated procedure is now high. Another problem is presence of stones. Stones are insoluble masses of crystallized calcium oxalate that are formed within the kidney. There are insoluble crystals. They are called kidney stones or renal calculi. Inflammations occurring to the glomerulus is called glomerulonephritis. So these are some of the problems that may affect our kidneys. So it is our duty to keep our kidney always healthy, and we have to look into the reasons why our kidney is malfunctioning, mainly because the food we take, the water we drink. So always use safe food and water. With these words, I will conclude my session and will meet you with another session, with another chapter next time. Until then, goodbye from all of us here. So thank you all. Thank you very much.